The epidermal growth factor, or EGF, is the primary messenger signal that starts the signal transduction pathway. EGF is released by cells and picked up by the cell itself or neighboring cells.
In normal EGFR, EGF binds to the receptor. When two monomers of EGFR receive the signal molecule, they dimerize, creating a dimer. Mutations of EGFR can happen in the transmembrane, extracellular, or intercellular domain. Point mutations in the transmembrane domain cause EGFR to spontaneously dimerize without the binding of a growth factor. Point mutations in the extracellular domain form cysteine bridges, leading to inappropriate dimerization. Mutations in the cytoplasmic domain activate EGFR without a growth factor binding. After dimerization, EGFR becomes activated and opens binding sites for ATP and epidermal growth factor pathway substrates, or EPS. Phosphorylation occurs and the P groups from the ATP are put on the tyrosine ends. EGFR is now ready to initiate the signal cascade.
In the signal cascade, cytosolic proteins recognize the phosphotyrosine containing binding sites through recognition domains such as the SH2 domain. A complex of GRB2, which contains an SH2 domain, and SOS binds to their receptor. A phosphate group is added to GRB2's side chain, changing it from a hydroxyl group to a phosphate group. SOS becomes activated, and the complex is translocated to the membrane in close proximity with RAS. SOS catalyzes RAS to release GDP, allowing it to bind at GTP and activate. GRB2 and SOS act as secondary messengers by initiating the RAS pathway. RAS activates a cytosolic kinase, RAF kinase. RAF phosphorylates and activates MEK. MEK phosphorylates and activates MAPK, or mitogen activated protein kinase. For reference, mitogen is a signal molecule that encourages cell division and mitosis.
MAPK can phosphorylate the transcription factor MYC, which is essential for cell proliferation. Co-expression of RAS and MYC leads to the initiation S phase, in which DNA replication occurs in preparation for cell division. The transcription factor MYC is inappropriately activated, initiating the S phase prematurely and accelerating the cell's life cycle. Thus, abnormal epidermal growth factor receptors are a primary cause of cancer through the activation of mitogenic pathways in the absence of a growth factor.
Tyrosine kinase inhibitors such as gefitinib are used to treat cancer patients. These inhibitors can block the binding sites for ATP in the intracellular domain of EGFR. EGFR can no longer be phosphorylated. Antibodies can block the binding site for EGFs in the extracellular domain. Without the improper transduction of the EGFR pathway, cancer cells will be unable to reproduce and die.